see here students on the image? Yeah, plant cell and animal cell. The fundamental unit of life, cell. Today let us continue the previous class continuation and let us recapitulate before going to today's topic. What did we discuss in the previous class? About the plasma membrane, diffusion and osmosis experiments. Let us discuss about study of cell wall. Cell wall is present in plant cell, fungi, bacteria, and it is absent in animal cell. Cell wall is a tough, ridge layer that surrounds the cell membrane to provide structural support and protection. Composition of cell wall in plant cell. Plant cell contains cellulose. Cellulose is a complex substance and provides a structural strength to the plant cells. Effect of osmosis on living plant. The living cells loses water through osmosis. This results in the shrinkage or contraction of the cell membrane away from the cell wall. This phenomenon is called as plasmolysis. Let us perform an activity before going to study more about cell wall in Royo leaf. Now let us study the study of plasmolysis.
Plasmolysis is the process of shrinkage or contraction of the protoplasm of a plant cell as a result of loss of water from the cell through osmosis. We can induce plasmolysis in the laboratory by immersing living cells in a strong saline or sugar solution to reduce water in the cell. Normally, people use Royo or Tradescantia plant epidermal cells for this experiment as their colored cell sap is clearly visible. Materials required: Royo leaf, forceps, glass slides, cover slips, compound microscope, needle, droppers, sodium chloride 0.1% solution and sodium chloride 5% solution. Procedure: take 2 glass slides and place them on the table. Take a Royo leaf from the petri dish. Fold the leaf and tear it along the lower side of the leaf. Using a forceps, pull out 2 small segments of thin transparent layer from the lower epidermis of the Royo leaf. Place the epidermal peels on both glass slides. Using a dropper, take some sodium chloride 0.1% solution from the beaker. Put 1 to 2 drops of solution on one slide. Using another dropper, take sodium chloride 5% solution from the beaker. Put 1 to 2 drops of solution on the next slide. Place a cover slip over the peel of both slides using a needle. Place the slides one by one under the compound microscope. Observe them under the microscope. Observation: after half an hour, we can observe that cells in sodium chloride 0.1% solution appear turgid, while cells in the sodium chloride 5% solution show plasmolysis. Conclusion: when plant cells are immersed in sodium chloride 5% solution or concentrated salt solution, water moves through the cell membrane into the surrounding medium because the water concentration inside the cell is greater than that which is outside the cell. Ultimately, the protoplasm causes shrinkage and assumes spherical shape. This is called plasmolysis. When a plant cell is immersed in sodium chloride 0.1% solution or dilute salt solution, the water moves into the cell because of the higher concentration of water outside the cell than inside the cell. The cell then swells and becomes turgid. Precautions: take the epidermal peel from the lower surface of the Royo leaf. Do not let the peel dry out.
Cell walls permit the cells of plants, fungi and bacteria to withstand very dilute hypotonic solutions or external media without bursting the cell. In such media, the cells tend to take up water by the process of osmosis. The cell swells, building up the pressure against the cell wall. The cell wall exerts an equal pressure against the swollen cell. Because of these cell walls, such cells can withstand much greater changes in the surroundings media than in animal cell.